Hello, I would like to thank you for the opportunity to present fRagmentomics, a NER package developed by my team to integrate cell-free DNA fragment features with their mutational status to support liquid biopsy interpretation. A liquid biopsy is a simple blood draw from a cancer patient. Within this sample, we can find cell-free DNA. The majority of cell-free DNA originate from dying hematopoietic cells. However, in cancer patients, a fraction of this DNA is derived from tumor cells. We call these specific fragments circulating tumor DNA. Compared to traditional tissue biopsies, liquid biopsy has transformed cancer care by facilitating cancer diagnosis, treatment selection, and disease monitoring. However, a key challenge remains. Because ctDNA is highly diluted within the total cell-free DNA pool, how can we effectively discriminate ctDNA from non-tumor cell-free DNA to improve clinical interpretation? To do this, we turn to fRagmentomics. fRagmentomics is the study of DNA fragment features. For instance, recent studies have shown that cell-free DNA fragments have different size profiles in cancer patients compared to healthy individuals. Here, the black curve represents the fragment size distribution for a colorectal cancer patient, which is shifted towards shorter sizes compared to the healthy individual profile in blue. We have also shown that cell-free DNA fragments carrying cancer-driving mutations are typically shorter than their wild-type counterpart, as you can see here, where we compared size of fragments bearing tumor mutations versus wild-type fragments in a pool of samples from Gustave Roussy Hospital. Finally, studies have revealed that fragment end motifs are emerging as powerful biomarkers for cancer detection. A very important point for our work is that we use capture-based gene panel cell-free DNA sequencing that allows us to detect both genetic mutations and analyze size profiles from the same data. To summarize, the analysis of cell-free DNA fragmentomics has many applications, but our study focuses specifically on tumor characterization. With dedicated therapies targeting specific mutations, it has become crucial to reliably identify tumor-specific mutations. To improve patient care and accessibility, we aim to use liquid biopsies to detect these mutations in the blood. However, this leads to a persistent challenge: the ambiguity of a mutation's origin. When a mutation is detected in a blood sample, does it originate from tumor or could it come from healthy cells? The solution we propose is to determine if the detected mutation is carried on DNA fragments that have characteristics of tumor-derived cell. This is why we develop fRagmentomics, a unified computational framework that integrates features of cell-free DNA fragments, like size and end motifs, with their mutational status. To start using the package, you need two main inputs: a mutation file and an alignment file. The mutation file lists the specific mutation you want to investigate. The alignment file, typically in BAM format, contains the sequencing data for your sample. You can search for both single nucleotide variants and indels. The output of the package is a data frame where each row corresponds to a single DNA fragment. For every fragment, the data frame provides its mutational status, for instance here mutated or wild type, along with its fRagmentomics features. So let's walk through the package workflow. The first step is to normalize the input mutation file. Then, for each mutation of interest, the package subsets the alignment file to the region around that mutation's position. For every fragment within this subset, it then extracts both the fRagmentomics features and the mutational status. This entire extraction step is parallelized to optimize performance. Here are two quick examples of the feature we extract. In this first case, you can see that only one read of the pair covers the mutation's position. At this position, the read carries the reference nucleotide. So its status is classified as wild type. We also extract the fragment length, in this case 167 base pairs, and the start and end motif you can see here. In this second example, we have a mutated fragment. Here, both reads of the pair covers the mutation's position, and both carry the alternate allele, so G. Therefore, the fragment, is classified as mutated. To conclude, fRagmentomics provide a robust and user-friendly analytical framework to explore cell-free DNA fragment features by integrating them with their genotype at user-specified positions. fRagmentomics is also, to our knowledge, the only existing tool that is able to study indels in cell-free DNA. We are currently applying fRagmentomics to cell-free DNA data from a large cancer cohort with several key objectives, like comparing these fRagmentomics features between mutations derived from cancer and those from clonal hematopoiesis. We have also performed a benchmark against existing tools to compare our results and performance. Ultimately, our goal is to infer the tissue of origin of cell-free DNA mutations and to develop a classifier that can accurately predict whether the mutation is tumor-derived or hematopoietic-derived. Finally, I would like to thank the entire computational oncology team, especially the co-author of this work, Elsa Bernard, Johan Prada, and Juliette Samaniego. And I also extend my thanks to the Precision Medicine Liquid Biopsy Program and our funding sources. Thank you.